At first glance, it looked like a plant, but under the microscope, it looked like a fungus. This is Prototaxites, a complex organism that does not fit neatly into either group. When it was first described in 1859, the plant interpretation seems straightforward. The fossils resemble large cylindrical logs preserved in rock, sometimes several meters long and standing upright. Some show ring-like banding that was compared to growth rings in wood. At a time when classification relied mainly on visible structure, a tall trunk-shaped fossil from the Devonian period was taken to be an early tree. More detailed study made that identification difficult to maintain. Prototaxites has no leaves, seeds, stomata, or lignified vascular tissue such as xylem and phloem. Tall plants depend on that internal system for water transport and structural support. Earlier work suggested the presence of an extinct polyphenolic structural biomacromolecule similar to lignin, but later chemical analysis confirmed that its composition is distinct from true plant lignin. Its growth pattern also differs from plants. It formed a single and branched column rather than branching from growing tips. In 2007, carbon isotope analysis showed that it did not build its tissues from atmospheric carbon dioxide through photosynthesis. Its carbon came from organic matter in the soil, which does not align with how members of plantae function. The fungal interpretation gains support from its internal structure. Under the microscope, the trunk contains interwoven tube-like filaments described as skeletal, binding, and generative types, resembling the construction of large bracket fungi. The isotope evidence indicating heterotrophy is consistent with organisms in fungi, which obtain nutrients from existing organic material. In 2026, researchers tested that interpretation using exceptionally preserved material from the Rhynie Chert in Scotland. They compared confirmed Prototaxites taeti specimens with fossil fungi from the same deposits. The fungi retain chitin in their cell walls. The Prototaxite specimens did not show comparable evidence of chitin under the same preservation conditions. Because chitin is a defining feature of fungi, its absence complicates a straightforward fungal classification. The same study also documented organized internal hubs and differentiated tube systems that did not closely match known fungal structures. Chemical analyses further confirmed that its structural polymers differ from both plant lignin and fungal chitin signatures. The evidence does not place Prototaxites comfortably within either group. It has the overall form of a tree but lacks plant anatomy and photosynthesis. It shares structural and metabolic traits with fungi but lacks confirmed fungal cell wall chemistry. Its exact identity remains unresolved, although many researchers now regard it as belonging to a previously undescribed, independent and extinct lineage of complex multicellular eukaryotes. It persisted for over 40 million years, from the late Silurian to the late Devonian, towering over low vascular plants and early terrestrial fungi before disappearing from the fossil record.